Hi, everyone. Thanks for joining us today. My name is Megan Sullivan. I'm a PhD candidate at the Yale School of the Environment, and I am the Research Spotlight Assistant for the Peabody, which means I have the pleasure of hosting these talks for you all. The Research Spotlight series is the Peabody's graduate student speaker series, and we use this series as an opportunity to spotlight different graduate students who do their research in connection with the museum's collections. Just a couple of housekeeping things to mention before I introduce our speaker. We have disabled the chat function for this webinar, so if you have any questions, you can use the Q&A to submit them, and feel free to submit questions at any time during the webinar. Sometimes we receive many more questions than our presenters have time to answer, so we do apologize in advance if we're not able to get to everyone's questions today at the end of the presentation. And I do want everyone to know that our program today is being live captioned, so you can turn on the captioning in the menu, usually in the bottom right of your Zoom window. So today we have Daniel Gaskell speaking for us. And Daniel is a PhD candidate in the Yale Department of Earth and Planetary Sciences. His work focuses on understanding the behavior of extinct microorganisms to reconstruct ancient climate. And his talk today explores the global quest to piece together Earth's climate history using septillions of fossilized microorganisms buried under the seafloor. He'll also give us a look at the fate of Antarctica in a warmer world. With that, I'll pass it off to Daniel.
Thank you, Megan. So this is the world as we know it. I'm showing here a reconstruction of global temperature since 1850. Blue is colder than average, yellow is warmer than average. As you can see, over the past 150 years, things have gotten a fair bit warmer. And as we move now from the past data here into a computer simulation of what we expect the future to hold, we can see it getting progressively warmer even still, and really quite dramatically so. With, excuse me. Yeah, so the key question I'm going to be asking in this talk is related to this idea of polar amplification. And that's simply the observation here that if you look at this plot, this is 2100, the equator has a couple degrees of warming. The poles have considerably more. The darker red here is sometimes showing upwards of 15 degrees C or almost 30 degrees Fahrenheit of warming. And this is a phenomenon known as polar amplification, and it matters a lot because the temperature of the poles is what we care about if we're trying to think about sea level and ice melt. That's where the ice caps are. That's what's going to control a lot of sea level rise. So I want to talk about this in several contexts, but maybe first to lay out a little bit, why does this happen? It might be the first question you have. There's a couple reasons for this. There's many different mechanisms. Some simple ones that are pretty easy to explain are latitudinal heat transport. So most of the sunlight is falling on the equator. That's where it's warmest. That heat has to get redistributed towards the poles. And as the climate warms, as we know, the hydrological cycle gets more intense. You get increases in storm systems, and that increases more of the heat transport from the equator to the poles. There's also something that we call the ice albedo feedback. So I'm showing a map of sea ice in the Arctic in 1989. When sunlight falls on ice, it's quite light, so it reflects off quite well and is not very much of it absorbed. If you fast forward to 2016, when the sea ice extent was significantly reduced, more of that sunlight's falling on the water, and it gets absorbed instead of reflected. So these are two reasons why we would expect that the poles might warm faster than the equator for any given amount of warming. So the key motivating question that I want to explore in this talk is, how strong is this polar amplification effect over long time scales? And I'm talking very long time scales here, as you'll see in a second. But this can give us some insight into how the climate system behaves as a general principle. And the outline here, I'm going to go through several different subtopics and kind of get back to this question to give you the necessary background to understand the research that I've been doing. But I want to start by placing this in the context of climate history. So what I'm showing here is a plot of global average sea surface temperatures over the last about 90 million years. So you have age on the x-axis within in millions of years ago, from going from left to right. Then you have global sea surface temperature on the y-axis. That's just a measure of global climate state. And I put a few points on here. The extinction of the dinosaurs is indicated, or non-avian dinosaurs is indicated at about 66 million years ago. Before that time period, things were quite warm. We're talking a climate of Connecticut being more like Florida. Much of the inner areas of the US were covered with an inland sea. And now as we move towards the present, you are here. It has cooled somewhat over the last 100 million years. And I need to emphasize, this is a million-year timescale. So all of modern climate change, and indeed all of the last ice ages of the last couple of 100,000 years, are all compressed into that little blip at the end. That's kind of why it goes down there. But the advantage of looking at a big timeline like this is we can think about the Earth system on a really macro scale. What does it do when it's warm, fundamentally? What does it do when it's cold, fundamentally? And it matters because here's kind of where we're headed, right? Here's the optimistic scenario. If we're able to keep carbon emissions under control, we see a couple of degrees of warming over the next a few centuries. That puts us back into a climate state that we haven't seen in a few million years. That still pushes ecosystems and nations beyond what they're prepared to deal with. If you imagine if, even just on a human scale, if the entirety of South France could no longer grow grapes, that would be a big impact. But we're also potentially looking at the more pessimistic scenario. Carbon emissions are unchecked, and we see tens of degrees of warming, really the warmest that we've been in about 40 million years. And that matters because 40 million years ago, there were no ice caps. That's enough sea level rise to submerge not just New Haven, but also Hartford. And the reality, of course, we're probably going to fall somewhere in the middle of here. And when we're trying to reconstruct what that's going to look like, we can use these past climate states as examples of how the Earth system behaves in these situations. And how does the whole planet behave when it's warmer? So we're going to do that using fossil evidence. So I want to introduce a concept here. This is what we call a proxy measurement. And here's an example. This is not an ancient climate example, but it's cool. Did you know you can estimate the temperature outside by listening to crickets? And that happens because their metabolism is connected to temperature. So you can count the number of chirps in 13 seconds, add 40 to that, and that gives you the temperature in Fahrenheit, approximately. It's a cool thing. You can try it on your next camping trip. This is a proxy measurement. It's not actually a thermometer. The crickets don't have like a little mercury bulb in them, but it tracks temperature in a consistent way, so we can use it indirectly as a thermometer. I mean, there's a lot of things like that in the fossil record we can do. And I'll talk about one of them in more detail, but here's kind of one of the key conundrums I want to talk about. So one of the other ways we can approach the problem is by doing computer simulations of climate. And this is what the IPCC uses. This is what a lot of working groups use to try to reconstruct this. Computer simulations will say, on average, over million-year timescales, the poles seem to be warming about 1.25 times faster than the global average. Here's some data, some preliminary data from organic proxies. And there's a couple of problems with these proxies. They're kind of a broken thermometer in some ways, but they give us an idea of what might be happening. So I'm showing here latitude on the x-axis from the South Pole on the left to the North Pole on the right, and then sea surface temperature on the y-axis. And there's a couple of different climate states that I'm highlighting here, right? So the modern curve is this black one. This is a warmer climate state about 45 million years ago. This is a very warm climate state about 55 million years ago. And what we're seeing here is an enormous amount of polar amplification. We're seeing a climate state in the warmest periods where the gradient from equator to the poles is almost flat, which is insane. Can you imagine, if the North Pole was like Florida, not just Connecticut? And the models really can't account for that. We don't know what in the world could be causing that. And so the question arises, are we missing something fundamental about the climate system, or is this particular proxy system just a bad thermometer? So we need a more systematic picture of this. And so that's what we're trying to do. So I want to introduce here the group of organisms that I work on, which are called Foramifera. And these are, they're about the size of a human hair, and they are plankton that live in the ocean. There's also ones that live on the seafloor. They form a shell out of calcium carbonate that fossilizes very well. So that's convenient for two reasons. One, they fossilize very well. There's a great fossil record of them. And two, they're forming their shell basically out of chemicals in the seawater. So we can use the chemical composition of the shell to tell us something about what the seawater was like when they grew. So I'm going to just geek out here about forams for a second and show you some cool micrographs. So these are forams. They have formed these little chambers on their shells. They live, it's a single-celled organism, eukaryotic organism that lives inside that shell. And they do all kinds of interesting things. So this is a species that has long calcite spines that stick out from the shell in all different directions, and they can use those for prey capture or other things. This is a species that actually has a group of symbiotic algae on the outside. So essentially it's running its own little algae farm. It puts the algae out during the day to photosynthesize, as you can see here, pulls them in at night and harvests the starches that the photosynthesis produced. So really like crazy cool organisms. They also have some other interesting, one of the reasons I love them is they do with a single cell stuff that you can only do normally with you think of a large bodied organism. Here's one that has this bubble capsule it's using for buoyancy somehow. I did a paper on this species a few years ago. They also have these pseudopodia, which are, if you remember how an amoeba works, they basically are extending parts of their outer skin, basically, into these long arms that they can use to pull themselves around, do all kinds of interesting things with. That's also part of how they construct their shells. Apologies for the quality of this video, but they can use this to draw food in that they capture from the environment around the shell. Some of them, though, actually eat things that are quite a lot bigger, which I think I have a clip of here. When we're dealing with them in culture, we actually feed them artemia, which are these brine shrimp. They're basically what you get if you've ever had sea monkeys. That's basically what these are. And here you can see there's a foram on the left that's captured a brine shrimp that is considerably larger than itself. And it's basically injecting its pseudopodia into the shell of the brine shrimp and sucking out its innards. And it can completely eat this thing in about 30 minutes. It's really dramatic to watch under a microscope. So those are Foramifera. They're very cool. But one of the other weird things they do, their reproduction strategy is to basically turn their entire body into little gametes and then explode, which you can see happening here. I love these, and I spent a lot of time working on the biology of them, but I want to talk about them today as a proxy reporter. So why would they work as a proxy reporter? So here's a diagram of the seafloor at the bottom. We have forams living in the ocean. You can imagine that as sediment accumulates on the seafloor, the forams are experiencing different seawater conditions. They're recording those in their shells, and then they're getting buried, forming these progressive kind of, I like to call them planetary tree rings. Because we can come along, we can use a drill core, pull them up, and then analyze them layer by layer to tell what the ocean and indeed the whole Earth system was doing at different points in time. And we do that with there's a big international project to do this in many different forms. So I'm showing here one of the drill ships that is used to do this, the Joides Resolution, run by the IODP, or International Ocean Discovery Program. And they go on cruises. I like to say, people say, oh, you're a paleontologist. Do you go out in the field and wear your safari hat and brush things out of the soil? Well, my field work usually consists of emailing the IODP and saying, can you send me a box of samples? And then they do. So a little bit of a different picture, but this is the project that's going on right now. All right, so how do we actually get temperatures out of these in particular? So there's a lot of different ways to get temperatures from forams, but the one that I want to highlight is the oxygen isotope temperature proxy. So here's how this works. Most oxygen in the world is oxygen 16, which has 8 protons, 8 neutrons, 16 total. A small percentage of oxygen is actually oxygen 18, which has 2 additional neutrons. And it turns out that the ratio of these 2 isotopes of oxygen tracks temperature in a couple of different ways. The first way has to do with the physics of how foraminifera build their shells. So basically, it's a little bit easier to form a crystal out of oxygen 18 than oxygen 16. And normally that difference is quite small, but the colder it gets, the more likely it is that the crystallization process will favor oxygen 18 and exclude oxygen 16. So the colder the water the foram grew in, the less oxygen 16 it takes up into its shell. The second reason is a little more complicated, but it's kind of cool to get into. So here's how it works. So because it's missing 2 protons, oxygen 16 is just a little bit lighter than oxygen 18. So it tends to evaporate more easily and condense more slowly. So when water evaporates from the ocean near the equator, a little bit more oxygen 16 evaporates than oxygen 18. And as that water vapor moves towards the poles, the oxygen 18 that did actually evaporate out tends to sort of condense out and rain out. So by the time the water vapor reaches the poles and it falls out as snow, it has significantly more oxygen 16 than seawater does. So because that snow is what forms the polar ice caps, during colder periods when the polar ice caps are larger, we're actually removing a lot of the oxygen-16 from the ocean and trapping it in the ice caps, which kind of changes the whole composition of the seawater. So there's various reasons for this, but we measure this process using a value that we call delta 18O. I won't get into that except to say that it's a measure of oxygen-16 versus oxygen-18. And as I just described, it can be used to infer temperature. So let's do some climate reconstructions. So I'm going to show a bunch of plots here. I will try to walk you through them, but a lot of this project is data collection. So what we've done, and when I say we, I mean myself and then several other collaborators in the Hull lab here at Yale and elsewhere, who I'll credit at the end, is to compile about 28,000 delta 18O measurements from literature. These are all over the world, different laboratories house them. A lot of these Foramifera, not so much the ones we use, but often are even in the collections here at the Peabody. I'm showing here kind of a timeline, so 95 million years of time on the x-axis, left to right, delta 18O on the y-axis. But this sort of data isn't super meaningful by itself, so let's actually dig into what it says about climate. So here's the simplest thing you can do with this sort of data. I'm showing two time periods, the modern and about 60 million years ago, and I've plotted these data by latitude. So on the left is minus 90 is the South Pole, positive 90 is the North Pole, 0 is the equator. And the higher the point here, the higher the temperature. So the thin black line that you see is what the temperature curve looks like in modern. And this thick black line is what you would reconstruct based off of the data from the past. And you can see that 60 million years ago, it was indeed warmer than it was today. And in fact, the poles had warmed a bit more than the equator, which is polar amplification, and that's what we would expect. Now, something you might be wondering if you're looking at this is, why are all these different colors here? And why did the lines not always go directly through all of the points? And this has to do with one of the tricky parts about using these data, which is that the chemical composition of a fossil is actually strongly affected by recrystallization after burial. So here I'm showing some electron micrographs of Foraminifera shells, showing on the left when they're fresh, and then they get progressively recrystallized as they're kind of sitting in the seafloor. And if you can imagine, you're reforming a crystal at the bottom of the ocean where it's much colder, it starts to kind of overprint this basic temperature signal that they're recording with the bottom water temperature. And so this is something that we have to be very careful of when we're dealing with these sorts of data. And what I'm basically showing here, the black points are our best preserved data. And then as you get kind of progressively more yellow, the data gets worse quality. So we're taking that into account when we do these kinds of reconstructions, as well as some other details that I would get into if it was a conference presentation, but you can ask about if you're interested. So I showed you some plots of particular time intervals, but can we actually do this better and think about it systematically? So this one's a little bit harder to wrap your head around, but I'll try to explain it as best I can. So what I'm showing here are the same data, same colors, excuse me, but instead of pulling out discrete time intervals, we're taking this entire data set, we're dividing it into the tropics and the Southern Ocean. So that's the polar temperature around Antarctica. And we're plotting it with bottom water temperature on the x-axis, which is basically a way of measuring global temperature based on the temperature of water at the bottom of the ocean, which is pretty much the same over the whole globe. And then on the y-axis, we have sea surface temperature. And so if you remember, when you look at these black points, these are the best preserved samples, if you recall from the previous slide, we see this shockingly straight relationship between global temperature and then the surface temperature in a particular regional band. So as global temperature increases, so does the sea surface temperature, but at different rates for the equator and the poles. And that's our polar amplification. So there's a lot of cool things about this, but one of the things that it indicates is that we actually can use this bottom water temperature, which we have a really good record of, to get a good estimate of sea surface temperatures at any given point in time. So here's what that looks like. This is kind of like that plot I showed of global temperatures at the beginning. We have age on the x-axis and millions of years ago. On the y-axis is sea surface temperature. And we have these different lines. So the long lines are our kind of inferred continuous record. The individual points are some of the data that we're using to construct that. You can see that it lines up quite nicely. And we can reconstruct both tropical sea surface temperatures and Antarctic temperatures through time. And you'll notice that the poles, when you go back 90 million years ago, it's much warmer than it is today. The difference between the equator and the poles is a lot smaller than it is today. But it's not extreme, right? We're not talking a completely flat, no difference at all, like some of the proxies were suggesting, which is interesting. So to wrap this up, right, turns out models maybe do make some sense. So the models were predicting 1.25 greater polar increase in temperature than global temperature. Some of these proxies were predicting almost 3 times greater, which is crazy. This larger, more systematic approach seems to be indicating, well, our number specifically is 1.39 plus or minus 0.15, which is fairly close to what the models had been predicting before. So, yay, forams. This is probably good news. I mean, our initial question was, how strong is polar amplification over long timescales? The answer to that seems to be about what the models predict, not quite as extreme as we maybe had potentially feared. So this is probably good news in the sense that we're not headed for the most extreme case, but there is still a world of difference between that initial 10 degree C increase scenario and 2 degree C scenario that I was talking about, a difference of climate states millions of years apart. So I'll close with this kind of idea. Something that I like about this research is that it's really emblematic of how climate science as a whole works. There's so many different people working on different pieces of this. We have people who are doing astronomical investigations, using satellites to measure weather patterns. We have people who are using climate simulations to try to figure out how the physics of the Earth system works. And then we have the practical people. You know, here's a job I would like, going out and tranquilizing polar bears from a helicopter to go look at their habitat and how their health is doing. And then people who do practical field work, just investigating, for instance, local plant ecology, which is something that Megan's worked on. So all of these pieces together make up a cohesive picture of how the climate system works. And often when we start investigating them, we start realizing that they match up pretty nicely. You know, we had some reasons to be concerned that maybe the models were wrong. Doing more research into it suggests actually just another line of evidence to confirm that the models are probably not too far off. And that's kind of how the field as a whole is working. And I'm glad to be a part of something like that. So thank you. Much of the work that I presented here was my own, but specifically done in collaboration with these excellent people here at Yale and a variety of other institutions that I've listed here. And we will take your questions.
Hi, Daniel. Thanks so much for doing that. That was a really interesting talk, and congratulations on getting to near the end of your PhD program right now. So one of the first questions that someone has put in the Q&A box is from Jason Marche, and he says, hi, Daniel. Scientists are concerned about increasing CO2. Isn't most atmospheric CO2 absorbed by the oceans and taken up by bivalves, thus strengthening their shells? Do climate prediction models reflect this CO2 absorption by the oceans?
Yeah, so this is actually a really interesting area of research that I've worked on a little bit. What the question is getting right here is that the largest kind of sink of CO2 is the oceans. When you have higher CO2 in the atmosphere, that changes, it increases the absorption by the oceans. So various things happen, right? So bivalves and other organisms are creating their shells out of calcium carbonate. And so in theory, it's a good thing to have more carbon in the oceans. But also increasing CO2 actually changes the pH of the ocean as well, which makes it harder for them to form their shells. And the change in pH is actually usually larger than the change in total carbon concentration, because there's already quite a lot of carbon in the oceans. So in general, we're more worried that CO2 would be a problem for bivalves, and particularly young bivalves that have very fragile shells and really need all the help they can get, which is why we've been seeing a lot of problems with, for instance, oyster fisheries collapsing recently. But the question about climate models is a very interesting one. So yes, there's actually several types of climate models. There are shorter term climate models, which are working on you set them up with a set of background conditions, you run them forward a couple hundred years and sort of see or a couple thousand years sometimes, and see what the climate state looks like under those conditions. So that's what's being used to do a lot of forecasting in the next couple hundred years. There's also the longer term climate models that are asking what happens to the whole Earth carbon cycle over time. And so we kind of have both of those. Absorbing CO2 into the oceans tends to be a process that occurs on more 1,000-year timescales, although the acidification effect can be quite rapid. And that occurs partly because you can just imagine the timescale of ocean overturning. basically the whole global circulation, how you get water from the surface to the deep and back to the surface, that takes about 1,000 years. So that's a longer time scale that we're working with. But yeah, that's a great question.
Okay, cool. Thank you so much. Next, we have Ellis Weisner. Have poles and oceans moved with the movement of tectonic plates?
Ooh, yes, they have. Do you know if this question is asking about the climate implications of that? I mean, I guess I could talk about that.
Yeah, so, sure, go for it.
Yeah, so one of the big things that's happened over the last 100 million years is that we have the breakup of Pangaea, right? So Pangaea is the supercontinent that exists during the age of the dinosaurs, starting, I'm going to say 150 million years ago. This is a little bit outside of my I don't work with the chronology of this very often, but we start to see that breaking up and forming, particularly the Atlantic Ocean opens up. So we get a separation of North America and Eurasia. And that whole process, there's a lot of debate about why it is that temperatures have been slowly decreasing over the last 100 million years. And so it's thought to have something to do with that tectonic process. The exact mechanism is a little bit unclear. It might have to do with the Himalayas rising. It might have to do with just the change in sort of surface area. It might have to do with biological responses to that whole process. But it's very actively debated. I actually have a colleague who's working on a paper where she basically summarized, here are about 15 different things that potentially could have caused this, most of which are tied in some places to tectonics. So yeah, that's a process. And it's also something that we have to take into account when we're doing reconstructions, because, I mean, just to make this even more collaborative than it already is, in order to know where, in terms of latitude, our samples are actually coming from, we have to use reconstructions based on people who are working with paleomagnetism and sort of continental drift to tell like where things even were in the past. Because the sample that was originally from the equator might have ended up now somewhere in the South Pacific or what have you. So yeah, these are all questions that are fascinating to work with.
Okay, that's really cool. I did not know that. Thank you. The next question we have is from Chet Serge, and they say, have you compared global temperature variation shown by the microfossils with the effects of the Melnikovitch cycles, including the variation in the tilt and precession, I think, or precession,
Yeah.
of the, okay, thank you, of the Earth's axis, and on global temperatures versus that claim to be caused by anthropomorphic CO2 generation?
Yeah, so for context, for those who aren't familiar with Melnikovitch cycles, basically the Earth, due to its orbital properties, has several cycles that occur on sort of 20,000 year, 40,000 year, 100,000 year timescales, where the axis of the Earth is actually wobbling sort of around in a circle. How elliptical the orbit is around the sun also changes. And it creates these kind of cycles of how much and how the sun is influencing Earth's climate. And when you look at shorter timescale records, there's a lot of people who've done work with Foramifera looking at the past maybe 100,000 years or a few 100,000 years, trying to notice that. And it's actually, it works surprisingly well. So the impact of Melnikovitch cycles on climate is precise enough that you can actually use it to date the samples, because you look at the wiggles and you're like, wow, that's exactly a 40,000 or 41,000 year oscillation, and it happens to line up perfectly with the Melnikovitch cycles. And so this is actually something called astrochronology. You can like use that as one of the different methods we use to date the samples. So yeah, that's something that occurs in the samples. The work that I've been doing is a bigger picture, right? So Melnikovitch cycles do take place over million-year timescales, but they kind of get lost in that long-term cooling trend that we saw over 100 million-year timescales. So that's something that takes place more over maybe 20,000-year scales, and it controls the ice ages in the last couple 100,000 years. The last part of the question was dealing with, anthropogenic forcing. So if you look at some of these climate simulations and also the basic physics of how the greenhouse effect works, and you overlay, like Melnikovitch cycles, right, they're astronomical, they're very regular, it's like a ticking clock. And so you can put them on a chart and say, where, if climate was just driven by Melnikovitch cycles, where should we be? And we were there up until about 150 years ago, when the temperature started rising. And we're no longer there now. And the only way to really explain that is by including in your models and in your statistics the increase in anthropogenic CO2. It really doesn't work any other way. So, I mean, I think at this point, the science of that is pretty straightforward. But by looking at that deviation, it actually gives us a much better sense of, is this natural variation? Because we can use the forams to reconstruct what the natural variation is, and then suddenly we're not seeing natural variation. So something else must be happening. Yeah, great question.
Great. There was someone who congratulated you on your fascinating presentation. Just wanted to plug that one. And then another person who asked, by 2200, will the year be summer all long? Summer all year long. Sorry.
Probably not. Depends on where you live. If you live in Florida,
yeah, in some places it's summer all year round already. So,
I mean, I suppose by definition, summer is related to the orbit of the planet, which isn't changing. So it will be summer. Summer will just be hotter, and so will winter. Yeah, I mean, I think that really depends on where you live and also on the trajectory of carbon emissions over the next 100 years. Because there is a world of difference between 2 degrees C of warming means things get disrupted, crop patterns move around, ecosystems are adapting. But fundamentally, the planet isn't becoming a barren wasteland. But I mean, there are other extremes that you could go to. And so yeah, I think that question is really going to depend on what we end up doing with carbon emissions in the next 100 years.
Cool, sounds good. We did have a question coming in, I think, from the YouTube stream. What limits population growth of forams? Does anything eat them?
Ooh, that's a great question. So this is bringing to mind a bunch of conversations that we've been having around the lab recently about how forams actually reproduce. It's fascinating. But yeah, I won't go down that rabbit trail. Yeah, on a large scale, what is controlling populations of just about everything is nutrient availability. So on land, the number of predators you can have is determined by the number of prey you have, which is determined by the things that the prey are eating, which is determined by sunlight and water and other sorts of features. So in the ocean, it's a similar kind of thing, except ultimately a lot of the nutrients are coming from stuff that's dissolved from the mountains and run down into the ocean, basically. So there are a lot of questions now about can we mess with that cycle? There's a whole project now, actually. One of the things that Yale recently got a large amount of funding to do is the Yale Center for Natural Carbon Capture Solutions, which is basically asking questions like, are there ways of increasing the amount of carbon that's being taken up by plankton in the ocean by either different fertilization methods or spreading rock directly into the ocean or what that might be. So a lot of interesting questions there. From an ecology perspective, yeah, a lot of it's controlled by nutrient availability, and that's why you get interesting patterns all over the world where some regions have a lot more forams than others. But in general, there's enough forams in any given one spot, you can usually do the research you need to do.
Yeah, thanks. No, that's super interesting that you say that carbon uptake stuff depends on, or could depend on, all of these different things in the ocean, and there are different techniques to do that. Because I, as a plant ecologist, terrestrial ecologist, I'm always thinking about that from the plant perspective, but it's really interesting to get that from the marine perspective as well. Our next question comes from Gargi Bhattacharya, and they say, hi, Daniel, thanks for raising this interesting topic. Other than changes in climate, what other factors are responsible for the extinction of species? And can you suggest some reconstructive steps and preventative measures to sustain ecological balance, avoid global warming and other threats? Big question.
What a great question. That's like a whole field of ecology there. I'll speak to the extinction thing at least. I'm not a conservation biologist, so I think that there are interesting perspectives we can learn from past ecosystems on that, but it's a little bit outside of my normal wheelhouse. But yeah, extinctions are fascinating. So some degree of extinction is natural. There are normal rates of background extinction, and there are things that, a virus comes along and wipes out all of a particular species, or mites come along and wipe out a particular species of tree, or what it might be. in the oceans, things are susceptible to climate change. In fact, some of the big extinction events we see in Foramifera seem to have been driven by ocean acidification. So you do get a natural increase in carbon from maybe a large volcanism event like volcanic eruption, creates the same sort of ocean acidification we're worried about now, and a lot of the forams die. Other factors that can control that, well, certainly the big one that comes to mind, right, is we were just talking about this today in my lab group. The mass extinction that killed off the non-avian dinosaurs was caused by a variety of factors, but ultimately the main precipitating one was probably an asteroid about the size of Manhattan smashing into the Earth, which completely disrupts so many ecosystems everywhere. And you can get events like that happening. You can also get more gradual things where something just shifts and the habitat's no longer suitable. So what we're worried about today, a lot of it is species have been adapted for certainly last couple 100,000 years, but often longer than that, to live in particular climatic bands, to have particular associations. We find this particular species of beetle in this particular forest. And if the climate changes more rapidly than that forest can move, then suddenly the beetle has nowhere to live. And that's the kind of thing that we're worried about. So on the adaptation side of that, I think that's a little bit outside of my wheelhouse, but there are certainly the easiest solution. Well, I say easiest. The easiest solution is don't emit as much carbon, but and then you start getting into questions of how to do that some other way. But yeah.
Okay, thank you. Another question from Suzanne Schrader. She says, how do the ice ages fit in with this? Are they shown in the mud samples?
Yeah, so I kind of mentioned the ice ages are all pretty much captured in that tiny little blue blip at the end of my plot there. This is really a macro scale look at how the whole Earth system behaves, which, I mean, I have to emphasize this again, it's kind of a cool way of looking at it because it lets you see through a lot of the smaller scale variation. But of course, the smaller scale variations matter for us. We live in the small scale. So yeah, how do the ice ages fit into this? So I mentioned Melnikovitch cycles, which are apparently the precipitating factor for ice ages. In the present moment, certainly for the next some thousands or tens of thousands of years, we're in what you call an interglacial period, which means that there is polar ice, but it's not covering large portions of the northern hemisphere. And we're expecting to be in that state for a considerable amount of time, just based on the Melnikovitch cycles. So in the next couple hundreds of years, I would say that the ice ages don't matter so much. In the scale of my analysis, they also don't matter so much just because it's such a macro scale. But when we start asking questions about how recently we've experienced a climate like this, then you start looking at ice age scales. I don't know if that answered the question at all, but hopefully that was helpful.
Yeah, I think it was very helpful. There's another question about forams here.
I love forams.
Excellent. So do forams have any particular preferred temperatures? And if so, do areas where forams gather in the fossil record suggest areas of high biological activity or optimal temperatures?
Yeah, so yes, so in the modern ocean, there's about 50 species of forams that live in the water column, and then considerably more species that live on the bottom of the seafloor. And the ones that live in the water column do seem to have very particular temperature preferences. And it's to the scale up. So if you imagine the water column, the surface is quite warm because there's sun. But as you go further down, you rapidly decrease in temperature. And so a lot of forams will live at a very particular, 10-meter chunk of that water column. And as looking at, for instance, the ice ages, if you look at the record of forams through the ice ages, they often don't show as large of a temperature variation as you would expect, because they're tracking that little bit of, they have their preferred temperature, they just always like to live there. And then on a macro scale, there's ways to control for that, which we actually did in this research, to make sure that we were getting real global temperature and not just like a little thermal niche. But yeah, we also see latitudinal effects. So during some periods of the past when climate has shifted abruptly, so one of the events that I worked on a little bit is called the Paleocene-Eocene Thermal Maximum, or PETM. This is a sudden global warming event about 56 million years ago, and probably volcanically precipitated, but we're not sure. And you see the forams moving to higher latitudes because the tropical ocean suddenly gets much warmer. And that's actually one of the ways that you can kind of track where they're going. Yeah.
Oh, that's really cool.
I will make one comment about biological activity. Interestingly, most forams like to live in low nutrient zones. And there's probably various reasons for that, but it means that they're spread out all over the planet. And so you can use them to track pretty much every part of the ocean. It's not just traditionally like highly productive ones like reef systems or upwelling zones. But yeah.
Okay, cool. And then I'm just going to ask one last question of my own. So you talked a lot about the Foramifera, and I was wondering, are there any other ocean organisms that function as proxy species, and like what makes a good proxy organism generally?
Yeah, that's a great question. So yeah, so forams are very widely used because they fossilize so well. Another organism that also fossilizes well are coccolithophores, which are essentially calcifying algae. They're algae that also form a little shell. So they're a lot smaller than forams, even smaller than forams, but they are also used for proxies. There's also a number of organic proxies that are based on organic material that's left behind by cells. So for instance, the plot that I showed where we're kind of saying, what's going on here? Why is the South Pole so hot? That was reconstructed using basically a proxy that's based on membrane lipids in archaea. So a group that's sort of related to bacteria. And there are various problems with those proxies, in part because they don't always just record temperature. Sometimes they record also productivity, which is interesting. We often want to know productivity. And also sometimes they're degraded by passing through the whole food web and into the water column. But yeah, there's a lot of different proxies, and often the best approach is to take several and kind of see how they line up. Oh, what makes a good proxy? Sorry, I didn't answer that part of the question. Very briefly, yes. What makes a good proxy? We can measure it. It has a good fossil record. Ideally, it's quite global. You can find it all over the place. And it has some property that we can nail down and say, this is just measuring temperature, or this is mostly measuring pH, or this is mostly measuring, the nutrient composition of the water, or something like that. And there's various systems for that. But if you can invent one, it's a great way to get tenure.
Oh, I bet. All righty. Well, I think this brings us almost to the end of our time today. I wanted to thank you so very much for putting together your presentation and taking the time to come and speak here today. So thank you, Daniel. And I just wanted to thank all of you out there in the audience for coming to listen to this talk as well. If you did enjoy this talk, there are going to be multiple other talks this semester in this series. Look out for our next speaker, Daniel Smith-Paredes, who's also an evolutionary biologist from the same department, the Earth and Planetary Sciences Department. And he's going to be discussing the evolution of vertebrate limb musculature on October 28th. And if you want to get notifications about when all of these talks are going to occur, you can follow us on social media and sign up for our mailing list. We have a mailing list that is available through the Connect box on our Peabody homepage. And we'd like to make sure that all of you know that about all of these wonderful online programs that we are able to offer through the museum. So thank you all again. And I really appreciate you being here today.